In this talk, we will learn about angiosperms, which are organisms that belong to the kingdom of Plantae. All plants evolved from ancestral green algae. The first plants arose around 470 million years ago and were the non-vascular plants that were also called as bryophytes. These include the current-day liverworts, mosses, and hornworts. Around 425 million years ago, vascular tissues evolved and gave rise to the seedless vascular plants, which include the lycophytes and monilophytes. Some examples of monilophytes are the current-day ferns. Around 360 million years ago, seeds evolved in plants. All plants that produce seeds can be grouped into two clades, the gymnosperms and the angiosperms. The earliest plants were non-vascular plants, and they gave rise to the vascular plants, which evolved vascular tissues. The evolution of vascular tissues is a key innovation. The vascular tissues had the ability to transport water and nutrients to different parts of the plant. There are two main types of vascular tissues. Xylem transports water and minerals, while phloem transports nutrients from the leaves to different parts of the plant. Plants that have vascular tissues are called as tracheophytes. The evolution of vascular tissues is a key innovation because it allowed the plants to become bigger and more diverse. Thus, it supported the adaptive radiation of plants. It also aided in the evolution of plant organs, which are stems, roots, and leaves. The evolution of plant organs resulted in the sporophyte becoming larger and longer living than the gametophyte. Hence, the diploid sporophyte is the dominant generation in all vascular plants. All vascular plants possess true stems, leaves, and roots, and evolved modified leaves called as sporophylls. These sporophylls bear spores which arise due to meiosis. The spores, which are haploid, give rise to the gametophyte. It should be noted that the gametophyte has no vascular tissues, and hence it is only the sporophyte that has vascular tissues. Vascular plants can be homosporous or heterosporous. One similarity between seedless vascular plants and non-vascular plants is that the embryo is formed within the gametophyte. Over time, plants evolved a protective covering around this diploid multicellular embryo. This allowed the embryo to form a sporophyte outside the gametophyte. The multicellular embryo with the protective covering and nutritional stores is called as a seed. Seeds are similar to spores that are observed in non-vascular plants as well as seedless vascular plants, in that they both can withstand harsh conditions. However, since the seed has a diploid embryo, it is more resistant to mutations than spores are, which are haploid. The seed is able to protect the embryo because of the seed coat, which envelopes the embryo. The embryo has an inbuilt food supply in the seed and can remain dormant for many years and hence wait for favorable conditions. One big advantage of seeds is that they can be transported to long distances by different media. These include wind as well as animals. Thus, the embryo can be dispersed to many different locations farther away from the parent plant. Seed-bearing vascular plants are also called as spermatophytes. All spermatophytes belong to two clades. The first clade is the gymnosperms, which evolved first. They dominated the terrestrial ecosystems during the Mesozoic era. One feature of gymnosperms is that they do not make any fruits or flowers, but they do make seeds. The second clade are the angiosperms, which dominate most terrestrial ecosystems right now. They are flowering plants, and the plants produce a fleshy covering that surrounds the seed, which we call as fruit. In all seed-bearing vascular plants, the sporophyte is the dominant generation. The seeds represent a dispersible stage in the life cycle of the plant. All seed-bearing vascular plants also show a miniaturization of the gametophyte. Let us take the example of a flowering shrub. The flower that is present on the shrub is much smaller compared to the overall plant. Within the flower, we will have the microsporophylls, which represents the stamens. In the anther part of the stamen, pollen grains will be made, and this pollen grain represents the male gametophyte of the plant. Similarly, when we look at the female gametophyte, they are made in the ovules, which are present in the ovary, which in turn is present in the carpel of the flower. Within the ovule, the female gametophyte is formed in the form of an embryo sac. Thus, we can see that both the male and female gametophyte are much smaller as compared to the entire plant. This is what we mean by miniaturization of the gametophyte. We can also observe that the male and female gametophyte are formed within the sporophyte, which is different than what we observe in non-vascular plants. In the case of seed-bearing vascular plants, the sperm are transferred to the ovule through pollen, and this pollen can be dispersed through agents like wind or insects or water. Angiosperms are the most widespread and diverse plants that are present today. They all can be grouped into one phylum, which is Anthophyta. Angiosperms are plants that produce flowers, and from those flowers, fruits arise. The flowers are used for sexual reproduction, and the sporophylls are present within the flower. The stamens of the flower are the microsporophylls, which will ultimately give rise to the male gametophyte, which is the pollen grain, and within the pollen grain, the male gamete will be formed. The carpel, on the other hand, is the megasporophyll, and within the ovule of the carpel, we will have the female gametophyte being formed, which will then produce the female gamete. Fruits are formed from the flowers after fertilization occurs. In the case of angiosperms, the fruit is a seed with a fleshy covering. There are two main types of angiosperms, the monocot and the eudicots, which are also called as dicots. Some differences between the two include the presence of one cotyledon in monocots, while dicots have two cotyledons. The cotyledons provide nourishment to the sapling once the seed germinates. The leaves of monocots show parallel veins, while the leaves of eudicots have veins that show a net-like or branching appearance. The vascular tissue in the stems of monocots are scattered throughout, while in eudicots they are arranged in a ring. Monocots show the presence of fibrous roots, while dicots or eudicots show the presence of taproots, where there is a main root from which different branches can arise. The pollen grain of monocots have only one opening, while in the case of eudicots, the pollen grain can have three openings. The floral organs, like petals, are usually in multiples of 3 in monocots, while in eudicots, they are in multiples of 4 or 5. These differences allow the easy identification of monocots and eudicots. All angiosperms produce flowers, which is a specialized shoot that has up to 4 types of modified leaves. These modified leaves are called floral organs or whorls. The outermost whorl is the sepal, which is also called as the calyx. It encloses the flower in the bud stage and hence protects all the inner whorls. Inner to the sepals are the petals. The petals are also called as corolla, and sometimes they can be brightly colored and have different shapes depending on the plant. Inside the petals, we can see the next whorl, which are the stamens. The stamens are also called as the androecium, and the stamens are the microsporophyll of the plant. They are the male reproductive organs and hence will produce the male gametes. The stamen is made of an anther and a filament. The innermost whorl is the carpel or pistil. The carpel is the megasporophyll of the plant and hence is the female reproductive organ. For a plant to produce flowers, the plant must attain sexual maturity. The flowers arise from undifferentiated cells called as meristems. These are similar to the stem cells that we observe in animals. There must be a transformation process where the vegetative meristems transform to inflorescence meristems from which the flower will arise. The inflorescence meristems then must undergo differentiation to form the different organs or whorls of the flower. All angiosperms are heterosporous and hence they make two types of sporophylls. Both sporophylls are present within the flower. The microsporophyll is the stamen of the flower and is also called as the androecium. The stamen is made up of two parts, the anther, which is a lobe-like structure, and the filament, which is a stalk-like structure. Within the anther, there are pollen sacs, which are also called as microsporangia. This is where the microspores will be formed. The megasporophyll is the carpel of the flower. The carpel can also be called as the gynoecium of the flower, and it can also be called as pistil. A pistil is normally used to refer to one or more carpels. When we look at the composition of the carpel, it has a stigma, which is at the apex, followed by a stalk-like style, and a bulging ovary. Within the ovary, ovules are formed. Ovules are where the megasporangia are. Male gamete formation begins in the anther, which is a four-lobed structure with a vascular bundle in the middle. In each of the lobe, there is a pollen sac, and thus, with four lobes, there are four pollen sacs, which represent the microsporangia. Within the microsporangia are microsporocytes, which are diploid cells and are also called as the microspore mother cells. These are the cells that will give rise to the microspores. The process of forming microspores is called as microsporogenesis. The diploid microspore mother cells, or microsporocytes, undergo meiosis to form four daughter cells, which is called as a tetrad. Each cell in the tetrad separates from the other cells to form microspores that have only one nucleus and hence are called uninucleate. Each microspore that is thus produced is a pollen grain. An exine forms around the microspore in order to provide protection. Following microsporogenesis is the process of microgametogenesis, which involves the formation of the male gametes. Each haploid microspore that's produced undergoes two rounds of mitosis. Thus, within the pollen grain, the first round of mitosis generates two cells, and in the second round of mitosis, only one of those cells undergoes cell division, and thus the pollen grain now has three cells. Hence, it is called as tricellular pollen, and two of those three cells are sperm cells. The pollen grains must be released from the anther, and this happens by the splitting and tearing of the anther wall. This process is called as anther dehiscence and allows the pollen grains to be expelled out from the flower. In angiosperms, the female gamete is formed in the carpel, which is the megasporophyll. The carpel is made up of the stigma, style, and ovary. Within the ovary, we can find many ovules. Each ovule has two integuments. This is different from what is observed in gymnosperms, where the ovule has only one integument. In each ovule, there is the megasporocyte, or the megaspore mother cell. The megasporocyte is the one that will give rise to the megaspore. A megaspore is formed through the process of megasporogenesis. In this case, the megaspore mother cell, which is diploid, undergoes meiosis to give rise to 4 haploid daughter cells, which are the megaspores. Out of the 4 megaspores, 3 will die and only 1 megaspore survives. The surviving haploid megaspore undergoes 3 rounds of mitosis. In the first round, 2 haploid cells are formed. In the second round, 4 haploid cells are formed. And then these 4 haploid cells undergo another round of mitosis to give rise to 8 haploid cells. This forms the female gametophyte. Now, 1 cell from each end comes to the center and forms the polar nuclei. The polar nuclei fuse to form a definitive nucleus, which is also called as the secondary nucleus. This is the only cell in the female gametophyte which is diploid. All other cells are haploid. There are 3 cells near the micropyle end, which comprise of 2 synergids and 1 egg cell. This egg cell is the female gamete. There are another 3 cells near the chalazial end, and they are called as the antipodal cells. Let us look at the structure of the ovule after megagametogenesis is complete. We can see that within the ovule, the female gametophyte is present. On one end, we have an opening called as the micropyle, and that is where the pollen will be deposited. On the opposite end, we have the chalaza, which is where the ovule is attached to the ovary. Within the ovule is the haploid female gametophyte. At the end that is close to the micropyle are 3 haploid cells, which include 2 synergids and 1 egg cell. On the chalazial end are another 3 haploid cells, which are called as the antipodal cells. In the middle, we have the secondary nucleus. For fertilization between the male and female gamete to occur, pollination has to happen, which is the transfer of pollen grains from an anther to a carpel. There are 2 main types of pollination. Self-pollination is when there is transfer of pollen from the anther of a flower to the stigma of the same flower or the flower present on the same plant. Cross-pollination, on the other hand, is when there is transfer of pollen grains from the anther of one flower to the stigma of a flower that is present on another plant. This is usually accomplished with the help of different agents. There are abiotic as well as biotic agents of pollination. The abiotic or non-living factors that mediate pollination include wind as well as water. The biotic agents of pollination are different types of animals. These can include invertebrates like insects, as well as vertebrates like birds, mammals, and even reptiles. Once the pollen grain is deposited on the carpel, it binds to the stigma of the carpel, which tends to be sticky. There is a compatibility check, and if the pollen is compatible with the stigma, the pollen gets hydrated. This hydration of the pollen is important because it allows for the degradation of the coat that's surrounding the pollen, which is the exine. As the exine degrades, the cytoplasm of one of the 3 cells present in the pollen grain, which is called as the vegetative cell, starts coming out of the germ pore to form a pollen tube. The pollen tube extends out of the germ pore, and the vegetative tube nucleus is present at the end of the pollen tube, while the male gametes, and there are 2 of them, are behind that vegetative tube nucleus. The pollen tube grows longer and longer and is able to pass through the style of the carpel and into the ovary. It finally reaches the ovule. It knows where it's going because there are chemicals secreted by the ovule that form a chemical gradient which is sensed by the pollen tube. The pollen tube enters the ovule and is guided to the egg by the synergid cells that are surrounding the egg. One of the male gametes fuses with the egg cell to form a diploid zygote. Another male gamete, which is present in that pollen tube, fuses with the diploid definitive nucleus to give rise to the endosperm, which is a triploid cell now. This is because the definitive nucleus is diploid while the male gamete is haploid. This process, where one male gamete fuses with the egg cell to give rise to a diploid zygote, while the second male gamete fuses with the definitive nucleus to give rise to the triploid endosperm, is called as double fertilization. Double fertilization is observed only in angiosperms. The diploid zygote that is thus formed undergoes many rounds of mitosis to give rise to a multicellular embryo. This multicellular embryo occupies the entire space within the ovule. The integuments will form the seed coat around this embryo. Thus, we say that the ovule is the one that actually gives rise to seeds in angiosperms. While the diploid zygote gives rise to the multicellular embryo, the triploid endosperm also undergoes cell division to provide nutrition to this developing embryo. In this figure, number 1 corresponds to the endosperm, while 2 corresponds to the zygote. As time progresses, we can see that both the endosperm and the zygote are undergoing cell division. As more time progresses and the embryo becomes larger and larger, it uses up all the endosperm, and hence the endosperm starts shrinking. By the end of embryogenesis, there's very little endosperm left. As embryogenesis occurs, the integuments fuse around the ovule and hence form a seed coat. The seed coat protects the embryo and is usually made up of 2 layers. The outer layer is called as the testa, while the inner layer is called as the tegmen. When we look at the embryo itself, it's also divided into different parts. The embryonic root is called as the radicle, while the embryonic shoot is called as the plumule. In the case of dicots, there are 2 cotyledons, while in the case of monocot plants, there's only 1 cotyledon. The cotyledons are the first leaves of the seedling and hence provide nutrition once the seed germinates. The base of the plumule is called as the epicotyle, while the base of the radicle is called as the hypocotyle. These are the different parts of a dicot seed. In the case of angiosperms, a fleshy covering is formed around the seed to form fruit. This fleshy covering is usually formed by the ovary. The fruit protects the enclosed seeds and helps with the dispersal of seeds by wind or even by animals. Based on how they are formed, fruits can be classified as simple fruits, which arise from single or several fused carpels, or aggregate fruits, where a single flower with multiple or separate carpels give rise to the fruit. We can also have multiple fruits where a group of flowers give rise to the fruit, as seen in pineapples, or accessory fruits which contain other floral parts in addition to the ovaries. Let us look at the life cycle of angiosperms. We'll start with the sporophyte, which is the dominant generation of the plant. The sporophyte attains sexual maturity and produces flowers. Within the flowers, we have the sporophylls. The microsporophyll is the stamen of the flower, and within the anther of the stamen, we have microsporocytes. The microsporocytes undergo meiosis to give rise to the microspore. The microspore undergoes maturation to form pollen grains, and within the pollen grains, there are 2 male gametes, or sperm cells. The carpel is the megasporophyll of the plant. Within the carpel, we have ovules, and within the ovule, we have megasporocytes. The megasporocyte will undergo meiosis to give rise to the megaspore. The megaspore undergoes mitosis to give rise to the female gametophyte within the ovule. For fertilization to happen, the pollen grain must be deposited onto the stigma of the carpel. The pollen grain then germinates to give rise to a pollen tube that passes through the style and ultimately will reach the ovule. It enters the ovule through the micropyle, and one of the male gametes will fertilize the egg. This results in the formation of a diploid zygote. There is a second male gamete, and that will fertilize the definitive nucleus to form a triploid endosperm. This process is called as double fertilization and is observed only in angiosperms. After fertilization occurs, the diploid zygote will form a multicellular embryo, while the triploid endosperm will provide nutrition to this developing embryo. A seed coat is formed around the embryo by the integuments of the ovule. A fleshy covering will be formed so that fruits are produced. The fruits will then leave the parent plant and be dispersed by different agents. When the conditions are right, the seeds will germinate to give rise to the sporophyte. The sporophyte will then attain sexual maturity to produce flowers. With this, we come to the end of our talk, where we learned about general characteristics of vascular plants, as well as the evolution of seeds and the advantages of seeds. We also learned about the general characteristics of angiosperms and how gametes are formed in angiosperms. We also learned about the formation of seeds in angiosperms and how fruits are formed.